My name is Rebecca, and this summer I was lucky enough to receive a USRA working with Dr. Workenhe in the Department of Pathobiology. My project was focused on engineering a herpes simplex viral vector for inducible cell death in cancer cells. One of the more challenging aspects of treating cancer is its ability to hide from the body's immune system and continue growing without any resistance. One of the avenues being explored as a cancer treatment is immunotherapy. What immunotherapy essentially does is stimulate or boost the immune system so that it can fight against cancer. One of the challenges with immunotherapy is that certain tumors are more immunosuppressed than others, and often there are not enough T cells within the tumor to respond to the treatment. One way to signal to the immune system and increase the amount of T cells in the tumor is to use oncolytic viruses, which are viruses that are naturally occurring or genetically modified to selectively target and kill cancer cells in the body. There are several clinical trials currently ongoing which are testing the effectiveness of oncolytic viruses as therapeutic tools for cancer. Oncolytic viruses are effective for this purpose as they are able to activate cell death within the tumor. There are certain types of cell death that are inflammatory and lead to the secretion of molecules which activate an immune response and increase anti-tumor immunity, even in tumors with low T cell counts. This type of cell death is termed immunogenic cell death, and there are several different types of this type of cell death, such as pyroptosis and necroptosis, and they can be activated during oncolytic virus infection, and they all have slightly different pathways. The goal of this project was to engineer an oncolytic herpes simplex virus which is able to induce a specific type of cell death, which leads to a high inflammation and immune response by expressing a pro-cell death protein when the drug doxycycline is administered. This targeted approach will allow for the activation of one type of highly inflammatory cell death and will be useful to determine the differences between types of cell death and will hopefully lead to a greater immune response overall in tumors. The virus was designed to have an inducible promoter, TRE type, which is driving the expression of the pro-cell death protein, which in this case is RIPK3 for necroptosis and NGAS for pyroptosis. RTTA is also being expressed in the cell. When there is an absence of doxycycline, the RTTA binds to the promoter and the cell death protein is not expressed, but when doxycycline is added to the system, RTTA binds to it, and the promoter is free to express the cell death protein and cause cell death. There is also a drug resistance marker which will allow only the viruses containing the system to be able to survive after the addition of the drug, allowing for selection of these viruses. So far, a donor plasmid was constructed by restriction enzyme cloning 5 separate fragments that were necessary for the system into PUC57, which is just a very simple base plasmid. Then once the entire system was inserted, it was transferred to a plasmid containing the UL26, 27 genes of HSV, or herpes simplex virus. The photo on the left is one of the gel electrophoresis images where the plasmid was being verified to have the proper insert and being prepared for the next step in cloning. I'll continue to work on this project as my honors thesis in the fall, and the next steps will be to engineer the virus itself by adding the donor plasmid into HSV infected cells and causing the virus to make mistakes and recombine with the new DNA, and then selecting for these recombinant viruses. From there, it will be possible to test the virus's ability to induce specific types of cell death within cancer cells or in vitro, as well as in mice or in vivo. I want to thank Dr. Workenhe and all my lab mates for their support this summer, and I'm really excited to continue this project in the fall. Thank you.